And you know bone marrow suppression means what? Anemia. If it is red cell line, susceptibility to infections. If it is WBC line, hemorrhagic tendency. If it is platelets. Okay. So all the cell lines they get suppressed. Okay. Sometimes one of the lines more than the others. True, but all of them get bear the brunt. You know. So bone marrow suppression means all three lines. Okay. Alopecia means hair loss. Okay. Hemorrhagic cystitis. It's an adverse effect which is seen if proper precautions are not exercised before chemotherapy. Okay. So cyclophosphamide is a unique example among all these drugs. Okay. Cyclophosphamide is just like a cluster bomb. Okay. What is a cluster bomb? Cluster bomb.
Cluster bomb.
B-O-M-B bomb. Yes. What is a cluster bomb? No. A cluster bomb is, I would say, if I simply say, it's an explosive device. It's a bomb which, when dropped off, generates smaller bombs. Okay, which then produce havoc wherever it is inflicted. So cyclophosphamide generates a number of active moieties, active groups. Okay, it's a large molecule when it's broken down, metabolized, and then generates a number of moieties. By itself, it's a prodrug. Okay, we'll see. There are a few other examples of prodrugs. So cyclophosphamide generates a number of active moieties. The ultimate therapeutic moiety or therapeutic group is phosphoramide mustard. Okay, phosphoramide mustard. But there are other groups. For example, acrolein. So acrolein produced by cyclophosphamide damages the urothelium or the mucosal epithelium of the bladder and leads to sloughing and hemorrhage. So it's an inflammation, okay, marked with hemorrhage. So hemorrhagic cystitis.
Only in the bladder or?
In the urinary bladder, because it readily is filtered through the kidneys and goes to bladder. So accumulates there, and if hydration is not enough, the acrolein concentration will be high, and there would be hemorrhagic cystitis, chances of it. So just for now, just remember this. We will come back to the topic once again at the end. Okay, so major uses in the cancers of breast, ovary, CLL is chronic lymphocytic leukemia. NHL is non-Hodgkin's lymphoma. Okay, non-Hodgkin's lymphoma. So it's a leukemia, and this is a lymphoma. So hematologic malignancies. Okay, busulfan also pretty acts the same way. Similar adverse effects. The use is only chronic myeloid leukemia. Okay. Cisplatin is not related to the classical alkylating agent group, but it has got a mode of action similar to this. It's a platinum compound, okay, rather than the alkylating compound. It's a platinum compound, which also has got a similar mode of action, similar adverse effects, but the toxicity, delayed toxicity is a little different. So it is more nephrotoxic and neurotoxic and ototoxic, okay, because of the heavy metals, platinum. Okay, so lung, breast, bladder, ovary, and head and neck cancers are the ones where it is used. You know, what are the most common cancers?
Where?
Okay, whatever you like. Okay, let's start from the males. Lung cancer, colon cancer. Okay, so colorectal. Okay, so lung cancer, colorectal cancer. Okay, and what about in females? Breast cancer, and then ovarian cancer, and then depends on the cervical cancer. Then depends on the location. Okay, sometimes you could also find lung cancer pretty common in females, especially the societies where smoking is prevalent. Okay, okay, so let's move on. Lomustine is a nitrosuria. Okay. And there is a long chemical name for it, but the abbreviation is CCNU, forget about it. We are already not interested. Pretty similar acute toxicity, but the delayed toxicity is minimal. Okay, so as compared to its forerunner, carmustine, this is lesser toxic in terms of the chronic toxicity, and the major use is in the brain cancer, brain tumors. So let's now see what DNA cross-linking is. Here you can see phosphoramide mustard, already I mentioned. This is the ultimate active moiety or the active drug. Okay, the active cluster bomb, you know, the active bomb released from the cyclophosphamide molecule, and cisplatin is here. You can see they resemble in their ability to carbamylate or to alkylate the DNA molecule. And here you can see this is a double-strand DNA helix, okay? And the special or a preferred place for the alkylating agents or cisplatin is binding to guanine, okay? Though it can bind to any other base, but the preferred base is guanosine, okay? So it binds to G, okay, especially where there are two G's together. It just binds there, so it's a GG linkage rather, okay? So it links to adjacent G containing nucleotides, and this is a covalent interaction. So once done, it's gone. Okay, it has to be repaired, right? But this is not the only target. There are other macromolecule-like proteins which are also target of the cross-linking. But the major anti-cancer mechanism of this is DNA cross-linking. Okay? Okay? Is it clear? Yes. Anti-metabolites, or the ones which are disrupting the purine synthesis, pyrimidine synthesis, nucleic acid synthesis, or folate metabolism, all of them together. They are anti-metabolites. You know that yeah, but you apologize, but what about you? I mean, yes, you. The distant one, yes. I see you very often talking to each other, you know. Wherever you sit, you just cross-link. Yeah?
We're G's.
So refrain from being phosphoramide mustard. Okay. Okay, so methotrexate, 5-FU, Ara-C, and 6-MP. These are the four representative compounds present in your drug list. Methotrexate is the folate analog. Okay, last time we already discussed how it works. It blocks the dihydrofolate reductase, which is an enzyme. It brings about the tetrahydrofolate form of the folic acid, so it prevents the activation of folic acid. The toxicity is mucositis and myelotoxicity because it's a folate analog. You can understand why. The major uses are breast, head and neck, non-Hodgkin's lymphoma, bladder, and bone. Okay. For you guys, you need to understand the reason. I mean, the uses. But I would be recommending that you make a priority list. For priority list, I mean, first you need to understand. You need to memorize the drug name. Second, you need to memorize the mode of action and major toxicity. Okay, if you are able to catch hold of these three columns, you are ready.
So,
Okay, the uses would vary. There would be other uses as well. Okay, so where I stop, don't push me to go further. Okay, so 5FU, pyrimidine analog blocks thymidylate synthase. It's a false nucleotide, binds to the enzyme while synthesis of the thymidylate, you know, the thymidylate is synthesized from uracil. Yeah, so uracil is the precursor of the thymidylate. Okay, so when it binds to thymidylate synthase, it blocks it so that there is no more thymidylate. Okay, so thymidylate acid is no more available. So the cells undergo thymine-less death. Okay, so without thymine, the DNA won't be complete, won't be able to work, and there will be thymine-less death. Okay, so if you are asked which one is the drug which produces thymine-less death, it's 5FU. Okay, and luckily this is the only pyrimidine analog which is in your list. So nausea, mucositis, myelotoxicity, and neurotoxicity. These are the prominent adverse effects. Uses breast, gastroesophageal, liver, colorectal, all of it. So all of these major cancers. Okay. So Arac or cytosine arabinoside or cytarabine, as we discussed last time, they are the same. They refer to the same molecule. Okay, the same drug. It's a purine nucleoside, and again the same way it forms the pseudonucleotide. And the toxicity is also pretty the same, but the uses are not these. The uses are different. Hematogenous cancers. Okay. Now, why this happens? It's a preferential lack of salvage pathway in lymphocytes and hematological cells, which is even worse in malignant cells. That's why the malignant cells bear the most burden of the loss of the purines, and that's why they are more effective in hematological cancers. This is just repetition of your biochemistry with the purine synthesis and purine metabolism. So AML is the acute myelogenous leukemia, ALL is acute lymphocytic leukemia, CML chronic myeloid leukemia, and CML blast crisis. You know, there is an acute exacerbation. CML, you know, goes indolent, goes slow, all of a sudden sometimes there is an acute exacerbation, okay, which resembles an acute on chronic sort of situation. So acute phase of the CML, okay, so that's called blast crisis. So CML blast crisis, okay. Now the third one is 6-mercaptopurine. The name indicates purine, okay. Mercapto, whenever you talk about mercapto, you are talking about sulfhydryl group. Mercapto, okay. So mercaptopurine means it is a sulfur-added purine, okay. Mode of action forming pseudonucleotide with the same myelosuppression and hepatotoxicity are the major effects. Yes.
Doctor, why the third one was useful for only hematological?
As I said, the hematological cancer cells, they are particularly deprived of the salvage pathway of purine metabolism. You know, purines are synthesized either de novo, means from the very beginning. I mean, the cells form the purines from the scratch, or they salvage the broken down purines either from the metabolism or from the food. Okay, so the salvage means I would say rescue or recycling sort of pathways. So salvage pathway is another major pathway for bringing in the purines, okay, maintaining the purine pool within the cell. So if that is deficient, then the cell, and if you block the synthesis of the purine, then which cells would be the most burden? The ones which are also deficient in